In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum, dear students. How are you all? I welcome you in Pakistan International School Types virtual learning class for biology. And this is lecture number 19 for second year. Dear students, we have started chapter number 17, nervous coordination. And in that chapter, we have seen the introduction of nervous coordination. Then we have seen receptors, analysis of information in the CNS, and then we have seen effectors. Then we have just checked out the neurons, structure of neurons and different types of neurons, and then we have seen reflex arc. Today we are gonna see very interesting as well as very important topic of this chapter, that is nerve impulse. So in this chapter we will see how the nerve impulse is generated and how it is conducted towards the next neuron. So let's get started. Nerve impulse is actually, what do you know about it? It is signal, or it is the information about what the stimulus we are feeling, what stimulus is going on in our environment, and how does it transmit from receptors to CNS, and then from CNS, like central nervous system, to the effectors. Actually, in technical terms, a nerve impulse can be defined as a wave of electrochemical changes. Wow, it's a different name. Electrochemical changes. How does it happen in our body? We will see in a few minutes. And that travels along the neuron from one end to another. So how these electrochemical changes are going to be happen, that we will see in the generation and transmission of nerve impulse. So before understanding this concept, or going to this concept, we will see okay, let's take a look on this short video. Okay. Okay, so when we zoom into the brain, you can see here, yes, these are lots of neurons, and you can see how the stimulus or nerve impulse is conducted from one neuron to another. Just take a look, closer look of this one. When there is impulse, so what happened, the charges move in and out of the neuron membrane. So you can see the positive charges move in, and that is recovered by moving out. So these are sodium. So like this, the nerve impulses move from one neuron to another, and at the last of this neuron, you can see here is a synapse. Like the ending of this, there are lots of vesicles in which neurotransmitters are there. These are the chemicals which take the signals or the electric charges from one neuron to another by these receptors, and like this the nerve impulses is transmitted. Okay, so let's see how this all is happened. Actually, the word electrochemical, that refers to the electric potential. So what do you mean by electric potential? Let's go to a physics for some time. So what happened here, electric potential means electric charges, or the thing can do the electrical work. So we can take example of battery, which is having charges in it. So when we just we can do lots of work with that battery, because that can move the charges from one place to another. So here also electric potential means distribution of charges and movement of charges here along the neuron membrane. So how does it happen, or how we can say that electric potential in a neuron is termed as membrane potential, and which exhibited in two different forms. We will discuss one by one. Number one is a resting membrane potential, and the other one is active membrane potential. Resting membrane potential is actually uh, you, uh, you can see when the neuron is at rest, when there is no stimulus going on. So when there is stimulus, definitely there is a disturbance in the charges, but here in resting membrane potential, no movement of the charges is there, no stimulus is done here. So what happened, you can take a look of this diagram. Like, let's zoom in. Okay, so you can see this is lipid bilayer, and in that you can see lots of gates here, lots of carrier proteins here. Okay, so this resting membrane potential is maintained by two type of charges. One is sodium ions and the other is potassium ion. These two charges maintain the membrane potential, whether they are in the rest or whether they are in the action. So here we can say that sodium, why it is the rest membrane is out from outside, it is positive, and from inside the cell it is negative. So this is the resting membrane potential, that neuron membrane is positive from outside and negative from inside. So this is the charge distribution on both of the sides. That is also called resting membrane potential, as well as polarized state of the membrane. So you can see this membrane, that this is sodium potassium pump. This sodium potassium pump work actively, means ATP is utilized here, and this is open for all the time, whether it is in action potential or whether it is in resting state. So what happened, uh, by this sodium potassium pump, continuously this pump just pushing out a 3 sodium ions outside, 3 here, and 2 potassium ions inside. So outside is 3 positive and inside is 3 negative. 3 positive, so one more positive outside, it means outside is having sodium and inside of the membrane is we are having potassium. So from outside it is more positive rather than inside. Okay, number two of this, like why it is positive from outside and why it is negative from inside, that this potassium is bound by lots of negatively charged proteins which are present in the inside this membrane, like in the cytoplasm of neuron. Proteins are negative, proteins are present. That's why from inside it is more negative. Okay, the next important thing is that there are leak gates for potassium ions. So potassium gradually it's leaking out of the membrane. So Here, when one potassium ion will move outward from this leak gate, or it is leaking outside of the membrane, so what happened? Outside the membrane, positive charge will be increased, and inside the membrane, positive charge will be decreased. It means outside is more positive than inner side. Although the concentration of potassium ions is 30 times greater in the fluid inside the cell, here this side, and the concentration of sodium ions is nearly 10 times greater in the fluid outside, like in extracellular fluid in the cell than inside. But because they are continuously moving out and inside, so they balance the positive charge outside and negative side inside, negative charge inside the membrane. Here you can see these gates. These are passive diffusion gates, and they are done by ion channel proteins, or facilitated diffusion is done like with the help of some proteins. So these two gates, this is sodium gate and this is potassium gate, these are voltage regulated gates. And these voltage regulated gates, they are open and closed when there is a stimulus. In the normal condition, when there is resting membrane potential, these gates are closed. To open them, they need some voltage, some energy, which they get from uh, what we can see the stimulus which is given to the neuron. So this was overall membrane, like resting membrane potential. And here is very important thing, that this is called polarized state. Polarized state means positive charges are up, and negative charges are inside the membrane. So just take a look on this neuron, and this neuron is actually now conducting a stimulus. So when the stimulus is sufficiently strong, up to the threshold level, actually just one thing is forgotten here, that is the resting membrane potential is minus 70 millivolt. We'll discuss in the graph as well. And when they reach to the threshold level, the stimulus is reached to the threshold level, like up to almost 50, minus 50 to minus 55. In some books it is written minus 55 millivolt. So reach up to this level, millivolt. So this is the threshold level. So reaches up to here. So what happened? The distribution of the charges will change. So how does when distribution of charges will change? So that is called active membrane potential, or that is called action potential. And by that, the charge distribution of the charges will be changed, or charges are disturbed here. So that state is called depolarized state. And what happened here, what we have talked about, sodium, potassium voltage gates are present. So when there is action potential, like there is a stimulus on this neuron, so what happened, the sodium channel will be opened by the voltage, or by the energy which they got from the stimulus. After opening the sodium channel, sodium start to rush into this neuron, and by rushing of sodium into the neuron, more sodium will be accumulated in the neuron, so there will be more positive charge. And by this state is called depolarized state. And now the stimulus is going to is generated here, and now it is going to conduct in the next direction. So what happened inside potassium and like comparatively, sodium ions are more as compared to potassium ion. That's why they got positive charge. Now from here, the stimulus is moved forward, as you have seen here in this diagram. It moved forward, but the okay, let's talk about its move forward. So then there are at several distances, at every place we are having sodium potassium voltage gates. So sodium gates are going to be open and sodium is gonna rush inside it. But what happened to the previous, because here the potassium gates are open now. This is we call depolarized state. But when this potassium gate will open at the back, so potassium will move outward, and yet sodium is inside, but potassium is moving outside. By potassium, more potassium moving outside, but they are the potassium gates move and conduct the potassium ions outside very slowly. But when the neuron just passes the impulse forward, so from back, potassium gates are open and it moves the potassium ions outside. By that, outside charges gets positive sign and inside a more negatively charged is there. This is called a repolarization. First is, we have seen that first is polarized state, then we have, first is polarized state, then we have depolarized state here, and then we also having a repolarized state. This repolarized state is very, very important to move the nerve impulse in forward direction, not in the backward direction. So as we know that nerve impulse moves from the cell body to the axon, so that they don't go to the cell body, they move forward into the axon. So okay, then the nerve impulse move more forward, and from back um, uh, it is what we say repolarization state. Now in repolarization state, all sodium gates are gonna close, and potassium gates are also going to close very slowly, until that uh, these sodium gates are going are closing very slowly. So that period is called a refractory period. In that, the neuron is going to take a rest, and then it is going to ready to just mm, to just have another stimulus. We are having, as we have just told you, that we are having here sodium potassium pump, as well as we are having potassium leak channels, and now they are continuously moving 3 sodium outside and 2 potassium inside. And from um, potassium leak channels, potassium is continuously moving like outward as well, outward. So with that, what happened, the positive charge, or that is called a refractory period, in that there is a recovery of sodium ions outward of this membrane, and potassium inside of the membrane. And then the action potential is moving forward up to the last of the neurons. As you have seen the structure of the neuron, this is, and now here is this here the action potential is moving this way, and in the last it is having lots of neurons and axon terminals, and it reaches to the axon terminals here in the last. So this is called a synapse, or synaptic end, or presynaptic node. We will discuss how does from this presynaptic node they move to another neuron, how does it move, how action potential moves towards this one, we will see in the next topic. So here, like the part of the refractory period in which the nerve of fiber is not able to give response to a stimulus to any strength, that is called absolute refractory period, prevents another action potential. So at the back, when there is a refractory period, so here near about the action potential, absolute recovery period is there, where this neuron is going to be ready to just achieve another action potential, but yet it is not able to do this one because due to the distribution of like potassium ions and sodium ions are not proper here. So dear students, we have seen here in this topic about polarized state, depolarized state, and then hyperpolarized state. So we can see here through um, this graph. So you can see membrane potential is described in millivolt here, and that is the time. Okay, so here we can say that this at minus 70, this is resting membrane potential, and you black line, you can see this one. This is a straight, this one. And what happened now? We are getting stimulus. When we are getting stimulus up to threshold value, like up to almost minus 50, so this is threshold potential by which our body can get action potential in the neuron. Then definitely the sodium ions start to move inside, and slowly this graph goes up and up to here plus 40 to plus 50. What happened? We got the peak of that action potential, and here it means the conduction of impulse is just transmitted towards the next, and from back we are having a repolarization state, that is like this potassium ions are potassium voltage gates are opened, and potassium ions move outward to repolarize the state. Otherwise, next stimulus can be received by the neuron, and like there can be disturbance in perceiving the information. So then this is hyperpolarization. So this hyperpolarization is the state when and this is also called the refractory period. Then potassium ions and sodium ions are taking like more because potassium voltage gates are very slow in the closing, so more potassium ions is getting out from these gates, out of the membrane, so they got hyperpolarized state. And afterward, from sodium potassium pump and sodium leakage gates, we can have the recovery period, and that is like again we got a resting membrane potential from the back of the action potential. So this can take actually 250 impulses per second. The refractory period lasts for about 4 milliseconds. So in 1 second, we can have 250 impulses almost. Okay, how this like all is happened, or it's how means speed up the nerve impulse is, how much it is. So like that is called velocity. So velocity of nerve impulse in axon, we know that in axon membrane and synaptic cleft, that is variable. Like this is a neuron, and here this is axon, and this is like these are the terminals, and from here to here this is another neuron. So we can see this is synaptic cleft, and through this synaptic cleft, that is the different velocity of nerve impulse as well as compared to the axon. One more thing, that velocity depends on uh, actually the length of the neuron as well, because this is too long and here the distance is very less. So the other thing is that myelinated fibers, this is number 2. So if these axons are having myelin sheath on that, we'll see like this is myelin sheath, we know that is insulator, so it does not allow the conduction of impulses, as well as does not having the sodium potassium gates or pumps here to conduct properly the charges up and down outside and inside. So the nerve impulses jump here because voltage gates are present on the nodes of Ranvier. These are called nodes of Ranvier, so they are present here. So this is called saltatory movement, like they jump from one another. So this is the Latin word, it means leap. So the cells move by the voltage or the charges move by leaping from one node of Ranvier to the other. And as we talk about non-myelinated sheath, if it is not having myelin here, so just nerve travels from 1 to 3 meters per second. But if it is myelinated, so it can be just take 120 meters per second speed. Okay, so okay, so next thing, another reason for myelinated fibers conduct faster impulse, that the myelin sheath acts as insulating, so it does not allow any charges to move, and also there is less requirement of energy because there is myelin sheath and they do not having voltage gates or other sodium potassium pump which needs energy for the movement of the charges. The another reason for that, that is the diameter of neuron fibers. Diameter of neuron fibers, like if the neuron fibers are thick, so they can conduct more faster impulse, because thin fibers having resistance, and resistance to it is having more resistance to electric current flow. Electric resistance to electric current flow is inversely proportional to the cross-sectional area. So if that is cross-sectional area is thin, so it will suffer more resistance or more disturbance to pass these charges. If it is having more diameter, so it means it just requires less resistance to pass the charges, so this will move the current very faster. Okay, so then they have to move from the synapse to another I told you the synapse to another neuron. So this time that will take, that is called synapse delay, because it just needs some time because here are the gates which can move this easily, but here is the empty space where they have to move slowly. So that is called synaptic delay. Okay, so dear students, I hope you understood this very interesting topic, and um, we will talk, inshallah, we'll continue this topic by structure of synapse and actually the synapse process, like how this nerve impulse conduct from one a neuron to another. So till then, Allah Hafiz, wish you all the best.